Now we can discuss that how DNA ligase it can be used under laboratory conditions or in vitro to create covalent DNA recombinant molecule or artificial recombinant molecules. So the DNA fragments, either it is the vector DNA or the source DNA, if they have been digested with, for example, EcoR1. And EcoR1, we have the idea that it produces 5' single-stranded overhangs or sticky end. So how in vitro or in laboratory we can create recombinant DNA molecule by using DNA ligase. So when the DNA fragments that are having sticky ends that have been generated by the use of EcoR1, if they are mixed together, तो mixing के बाद due to complementarity between the ends of two DNA molecules, तो वो आपस में anneal करेंगे, join करेंगे. और joining के बाद जो nicks या gaps रह जाते हैं, how it can be breached in vitro by using DNA ligase. DNA ligase, it works well at 37 degrees Celsius, that is at body temperature. But under in vitro studies, it has been observed that this temperature it is very high, because if we digest the DNA with EcoR1, the single-stranded sticky ends, they are only 4 base pairs, like AATT, that are not enough to resist the denaturation. To overcome this problem, one can join the DNA molecules at low temperature, like 4 to 15 degrees Celsius. Now we can observe that how DNA molecules from two different sources that are digested with EcoR1, how they can be ligated in vitro by the use of DNA ligase. So this diagram, it showed the DNA ligase to create the covalent DNA recombinant or artificial DNA molecule. So in first diagram, it indicates the vector or the plasmid that has origin of replication here, and then target sites for different type of restriction enzymes that also include EcoR1, and a selectable marker for antibiotic resistance, and here it is tetracycline resistance. Now this enzyme, it can be digested with an enzyme like EcoR1. So that it will produce 5' single-stranded protruding or overhangs or unpaired bases like AATT. In second diagram, we have the foreign DNA or our gene of interest that we want to insert in the plasmid vector, and it is also digested with EcoR1. And this foreign DNA, it may also have different cleavage sites or target sites for different type of restriction enzymes, and of course it also contains site for the enzyme EcoR1. So it is treated here with EcoR1 restriction endonuclease, and the foreign DNA it is fragmented depending how many restriction sites for EcoR1 are there. So it also contains sticky ends having AATT overhangs, AATT. So these sticky ends, they are complementary with the vector DNA. Now here we can face problem that if the DNA concentration it is very low, either the foreign DNA or the vector DNA here, then the transformants or recombinant DNA molecules that will be produced, they will be lower in number, because होता क्या है कि ये प्लाज्मिड जो है ये सेल्फ लाइगेट कर जाएगा इन द एब्सेंस ऑफ फॉरेन डीएनए अगर हम लो कंसंट्रेशन डीएनए की यूज़ करें. तो हाउ इट कैन बी ओवरकम वी विल डिस्कस लेटर ऑन एज वेल, बट दिस प्रॉब्लम वी कैन ओवरकम अगर हम फॉरेन डीएनए की या प्लाज्मिड डीएनए की हाई कंसंट्रेशन यूज़ करें. If we will mix these two DNA populations like foreign DNA and plasmid DNA, then because of complementarity, so they will join together. But it also have nicks or gaps at some points like here and similarly here as well. Now how these gaps can be breached? So what will happen that after creating this artificial recombinant in vitro, then these gaps they are breached by DNA ligase. So it will create recombinant plasmid. Now this recombinant plasmid that we have synthesized in vitro, it is ready for transformation. So we can transform any suitable microorganisms. This plasmid, it contains a selectable marker which is tetracycline. So whenever we will introduce or transform the suitable host microorganism, we will grow microbe on those media that have tetracycline antibiotic. So only those microbes that are recombinant, they will be able to grow on that tetracycline medium. So after transformation, this recombinant plasmid will multiply inside the host organism independently from the main chromosome. So we can produce number of copies of our recombinant molecule after introducing into suitable host organism.